Good morning, guys. Happy Thursday. I am home on a personal day today. So you have Mr. DeSetter with you today, and I am creating this video to walk you through what you're going to do in class today. So, Mr. DeSetter, I'm going to walk them through the instructions, and then if you want to pause the video when I say pause, and then let them complete the task, and then play again. So the first thing you guys are going to want to do is check your seeds. So if you put it under the plant stand, look for your hour. If you put it in the dark, then it's in the cabinet in the back of the room on the clock side underneath where it says goggles and scissors. So if you open that cabinet, there's blue trays, and be careful, the petri dishes can tip, but there should be blue trays there that say 5th hour AM or 5th hour PM. So find those and take some data down, and you're going to put your data in the data tables. So you might consider taking pictures of things. A lot of you chose as your dependent variable just counting the number of seeds that have sprouted, but you're also going to need to take some qualitative data. So quantitative is any data with numbers, which you could get through counting. Qualitative would be numbers like characteristics that are not numbers, like colors. So take a few minutes and look at your seeds, create a data table in your elephant poop lab slides, and record your data. We'll look at these again next class period. If you may decide that, man, my paper towel looks dry, and we didn't really want our paper towel to dry, so we're going to add more water. If that is something you're going to do, just be careful that you do the same thing to each of the groups. So go ahead and pause the video, Mr. DeSetter, and allow them about 5 minutes to do their checking of their seeds. Okay, you've checked your seeds. Great. Hopefully you're starting to see results, and you're being able to answer questions like, do seeds germinate better in the light or not in the light? Is light necessary for seeds to germinate? And then within that, is there a benefit to a seed going through an elephant's digestive tract? You don't have to make those conclusions yet. We're going to take another data set, but that's where we're headed to. So today in class, we are going to do two things. We are going to do a seed pre-assessment, and then that will be individual. And then we're going to do the plant growth lab, and that will be in your teams that you sit with. So you're in a big team now, but the teams that you sit with. Okay, so at this time, what we're going to do is we're going to complete the seed pre-assessment on the classroom, and it looks like this. And I just want, what you're supposed to be doing is think about what you know about seeds and what you think about seeds. So go ahead and head to the classroom and open it up. And on this slide here, it says describe what you think is occurring in each one of these stages. So what do you think is going on there? Give a little description, and then answer the questions as you go. Okay, so I would give them about 5 minutes to complete this. So pause the video and complete that. Okay, so if I can summarize what most students say, they say, I think that the plant is growing roots and leaves, and it's growing more leaves, and it's growing even taller leaves and more roots. And a lot of students even say at stage D and E, maybe the plant's starting to go through photosynthesis. So what students often say on this slide is, what do plants need to grow? They put a list like carbon dioxide, water, light, nutrients. And then this question stumps them. They're like, well, what is the source? Where does this come from if the seed starts growing under the soil? So they get that the nutrients in the water can still come from the soil, but they start to question things when they're like, well, wait a minute. How does a seed get sunlight if it's under the soil? So that's kind of confusing to students, and maybe that helps you address a misconception that you have, or maybe it challenges your thinking. So this is a pretty cool slide. It shows you the parts of a seed, And if you look, there's kind of like baby roots, there's baby leaves, and then there is a seed coat, which we're wondering if the elephant digestive tract affects or doesn't affect. And then the bulk is the cotyledon. And if you look over here, the cotyledon stores starches, which are carbohydrates and proteins. So based upon the information, how does your seed start growth? Most of you probably thought it starts growth by using the energy stored in the cotyledon. And then once it emerges from the soil, how does it continue to grow? Most of you are probably starting to think photosynthesis. So that maybe challenged your ideas a little bit, that the parent plant actually puts a bunch of stored energy, stored carbohydrates or starches in the seed that enable that plant to grow. Okay, so that's our pre-assessment. You can close that out. Now we're going to go on to the plant growth lab. Go ahead and have one person in your team share this with everyone else in your group. So you might want to click pause and give students an opportunity to share it with, it should be a group of 2 or 3. Okay, now that you've shared out, I'm going to walk you through it. What we're going to do today is, in a non-COVID world, we'd be looking at plant roots and plant leaves under a microscope. But COVID, we're just going to look at pictures of them. And what you're going to try to answer is, is there more growth occurring in the roots or in the leaves? And what does growth even look like inside of a plant? Okay, so go ahead and complete your pre-lab questions. You guys don't need to skip this because you got to see this. So go ahead and complete your pre-lab questions. And actually, you can probably complete the pre-lab questions when I'm done going through this if you want. Okay, so I'm gonna go ahead and keep moving. So what are we gonna look at today? Where does more growth occur, in the root or in the leaves? So on to slide 6. So how am I gonna know if I look at cells, if they are making new cells or if they're not making new cells? Well, let's apply some words to this. In the diagram on the next slide, you will see plant cells taken from a sample. When the nucleus is present, we say the cell's an interphase and it's performing its normal job. So if we look down here, the question is, what is a cell doing during interphase? It is doing its normal job. So whatever that is for the plant, maybe its job is to do photosynthesis. Maybe its job is to help take up water. Maybe its job is to move sugars around the plant. Just doing its normal cell job. And then the next question, how do you know if a cell is an interphase? It says that when the nuclear membrane breaks down and chromosomes become visible as dark line, we say it's endomitosis. So you can tell a cell's an interphase if it has a nuclear membrane still, and you can't see any of these dark lines known as chromosomes. Okay, so I'm giving you the answers to these 2 as we go. All right, so now let's practice a little. I think it helps if you can see some of these in action. So here we have a picture of a onion, a regular onion, and it is zoomed in 400 times. These are our light microscopes in the classroom usually allow us to see. And what we see here is a bunch of cells. And what's crazy is it doesn't matter if you look at your cells or if you look at plant cells or cells of an elephant or bacteria. When you zoom in on any life, you see these little cells. And all cells go through the same process called mitosis to create new cells. That's pretty amazing. And that's evidence of the unity of life. That the fact that we all use the same process to make more cells, and that we're all made up of cells, and that we all have nucleuses. That's pretty crazy. Okay, so what we're going to try to do is we're going to try to figure out how many cells are in the process of dividing, called mitosis, and how many cells are just doing their normal cell jobs. And that can give us a measure of how much the tissue is growing. Lots of cells in mitosis, lots of growth. Lots of cells in interphase, the organ really isn't growing. Okay, so let's practice. Are these cells in interphase or are they in mitosis? So Anytime you can see a circular nucleus, that's interphase. So, circular nucleus, circular nucleus, circular nucleus. These three, if you move this to the side, and you can do this on all of yours, guess and check, are an interphase. All right, go on to this one. Can I see a circular? No, I can't. I see these black-like things, kind of look like, I don't know, little pipe cleaners. Oh, that's mitosis. The DNA has condensed into chromosomes. That definitely looks like thread-like structures. That's mitosis. That doesn't look like these. Looks different. Looks like it's starting to be thread-like. Yep, you got it. That's mitosis. This one over here, well, it doesn't look like these, where I can even see the nucleolus. I don't even see a nucleolus, so I'm guessing that's mitosis. Yep, mitosis. Okay, so the question at the bottom, how can you tell if a cell's in mitosis? You cannot see the nucleus, you can't see that little circle, and you start to see these little thread-like things. Okay? All right, so that's the answer to that question. All right, so now you're gaining some confidence in how to look at cells and tell if they're in interphase or mitosis. So now we're going to go a little deeper. This lab is not only going to ask you to figure out if cells are in mitosis, but what stage of mitosis cells are in. And you've probably learned about these stages in middle school. There's 4 different phases that are easily recognizable in mitosis: prophase, metaphase, anaphase, and telophase. So PMAT. And then the last stage you aren't going to have to identify, but it's called cytokinesis. All right. Here's your diagram that will show you what the phases are. So again, this is mitosis. This is when a cell is making a copy of its genetic material and then it's dividing in two to create new cells. Why would you do this? For growth, if you're growing, or if it's a full-grown organism, for repair or just replacement. So in prophase, what you see, these purple structures are the chromosomes. You see that there's chromosomes present. There's no nucleus here. The nucleus would have been that white dotted thing. It's disappeared. And the chromosomes are in no particular order. In metaphase, they're lined up in the middle. In anaphase, this chromosome gets torn apart, and you see there's half of the chromosome over here and half of the chromosome over here. And then in telophase, those chromosomes start to become thread-like again. So these are the phases you're going to look at identifying. Okay, let's practice some of these together. So this one, what do I see? I already know this is mitosis because I can see the thread-like structures, and they kind of look like they're all lined up in a line. That's metaphase. Oops. So you can move I would guess, and then move the boxes. All right, this one, they are on opposite sides. They're being pulled apart. That's anaphase. These 3, we already recognize that they have a little nucleolus, that little dot in the middle, and then they also have a membrane, so we already knew those were interphase. This one down here, I can start to see thread-like structures. I can still see the nucleus a little bit. That one's probably pretty close to prophase. I will tell you, I think prophase is one of the hardest ones to see, but what you'll see with prophase is that the structures, the DNA in the nucleus starts to become thread-like, but in no organized manner. In a line, metaphase, a part, anaphase, and then this one is telophase. So it doesn't have a circular nucleus. You can see the chromosomes are all scrunched in that middle. They're going to start to move into a nucleus, but that's not present yet. Okay, now we're ready for the real thing. So first thing we're going to observe is roots. How much cell growth is going on in roots? All right, so here's what you're going to do. We are looking at onion roots. So here's a picture of what the bulbs of the onions look like. Here's their roots. We're looking at one of these little root hairs. If you could see it under the microscope under 100 times, this would be the root hair. And then we're zoomed in even more, and you can see this. So the slide you're going to be looking at in slide 13 is where you see it magnified the roots of an onion magnified 400 times. So what parts of the cell can you see? Well, I'll help you with this one. You can't see much. All that you can see under the microscope is you can see its cell membrane or cell wall, because plants have both. You can see a nucleus, which is the circular thing, and you can see chromosomes sometimes, and that's it. What pattern do you notice? I can see those in all of them, besides the chromosome. And the nucleus looks dark. The chromosomes look dark. Those are kind of patterns you might notice. All right, now that we've looked at it, now let's begin our counting. So on slide 14, here's our root tip image. You're going to count the total number of cells. Now, work harder, work smarter, not harder, right? We're not going to actually count all these cells. We are going to instead just do the area. So you're going to count the cells across the top for the length, count the cells across the width, and then multiply length times width to give you the area, which could tell you the total number of cells in that space. Okay, so go ahead and pause, count, and then share your numbers with each other and see if you get something pretty close to each other. Okay, now that we have the total number of cells, now we're going to do something a little different. Now we're going to count the number of cells that are in each phase. The phases we're going to count are interphase, prophase, metaphase, anaphase, and telophase. Okay, so again, I would do a sample count. So, like we did with the elephants, we didn't count the whole thing. We didn't count all the elephants. Instead, we did a sample transect, and then we can multiply out. So here's an example. I'll show you an example of how you could do that. You could split this in half, like I've done with the orange line, and count the number of cells in each phase. When students did it yesterday, some students were using circles to show that they had counted those. So I saw someone putting like a blue circle on all the cells they were doing as interphase. You can use that method if you want to. So you're going to count the number of cells in interphase in this grouping up here, and then multiply that by 2, assuming that this sample is pretty similar to this. You're going to do the same thing with prophase. Find the number of cells in prophase. So if I'm helping you out with this one, because this is one of the harder ones, I would say this is a cell in prophase. That's an example of a cell in prophase. And if I'm looking at any of the others, this one's really early prophase. So there's probably only 2 in prophase in those upper ones. That's the hardest ones to see. And then you're going to multiply that number by 2. So we have 2. Your total number for prophase would be 4. Do the same thing for metaphase, anaphase, and telophase. Then to find the percent, remember percent is part divided by whole multiplied by 100. So if I take the number here for prophase and divide that by the number I got in the previous slide and multiply that by 100. That will give you an idea of how many cells are in the process of making the roots grow. You're going to do the same process for the leaves. Okay, so you might divide this up in some way. On the leaves, I do not give you the line of where to divide it. You can divide it in any way that makes sense to you, in half, in thirds, in quarters. How you multiply it out would then be different. And then your final CER that you're going to do 1 per group of 2 or 3 is you answering the question, which organ, the roots or the leaves, because they're the organs of the plant, is going through the most cell growth. So I would think you would have at least 2 numbers to compare for your evidence. And you have to think, what number should I compare for the evidence? And then how does this evidence support my claim for which one is going through the most growth? And then you have some analysis questions after that. So now is the time for you to begin counting your roots and your leaves. And what you don't get done in class today will be homework for your team. Okay, that's it. I hope you are able to understand what to do. And if you have any questions, feel free to email me. Okay, that's the end of the video.